Welcome to the Nervous Breakdown podcast. This is our third episode investigating the complement system.
Let's start off with what is the complement system?
Well, the complement system is made up of proteins that aid the immune system in pathogens.
What are some of the what does it really try to do and achieve within this innate immune response?
Well, the complement system has many functions, namely three. The first one is opsonization, which is a form of tagging pathogens for cell recognition. The second is cell lysis, formed through something known as the MAC attack complex, the membrane attack complex. The final one is immune cell recruitment, being able to communicate with other cells to come. What are these three mechanisms? Let's explore them in detail.
So the complement system acts in sequential pathways. The final function of this system is cell lysis through inserting the membrane attack.
So, C3b will first bind to the pathogen. This will then bind the C5 convertase, which will then be cleaved into C5b. C5b will associate with C6 and C7. C8 can then bind to C7, which will polymerize to add on C9, which will then, as a whole, form a pore in the pathogen surface and will lead to cell lysis. Negative back. So,
The thing here is you have your classical and you have your alternative. It's really for envelop bacteria that have cell membrane. If this isn't really present, such as things as non-enveloped viruses, this process is non-functional. It's completely pointless. Can't form a pore in no membrane. So you need specific membranes. Also, cell walls on bacteria, and you don't really have this fitting in this pore into their membrane. So it's very specific for bacteria with easy access to the cells through the cell membrane, and also your enveloped viruses, which kind of brings about differences in the different types of pathogens. But also, when you see deficiencies in different complements, it means you're liable for infections or diseases via a specific array of pathogens. Yeah, it wouldn't really be able to undertake all these functions because all these functions need to happen on a membrane of pathogen, on the surface of a pathogen. So if it doesn't really have this membrane or doesn't have this medium to really attach to and kind of build up, you can't really target this pathogen with, let's say, a complement system. This is why you need other types of immune responses for this purpose.
any specific immune responses?
Active immune response. Um, so yeah, your adaptive immune response, your T cells, your cytotoxic T cells, really guiding destruction of that pathogen. Different manner.
Okay. I think something very important to discuss is the regulatory factors of the complement system.
Yeah. So once you start thinking about it, if you have your alternative pathway, which is just spontaneously occur, would this not affect your cells, right? This could happen where within in the bloodstream, within tissues. So how can you really stop this system from attacking your own tissues? And this is where you have regulatory factors where properdin positive. What it does is it stabilizes the 3B BB complex. So it's able to stabilize this forming. So you're able to form your C3 convertase, stabilize the C5 convertase, so in MAC formation. You have also which is a serine protease, and it degrades E4B. And what this will do is inactivate these complement factors. So even if they bound to your cell surface, they're non-functional. They can't associate with C2. They can't associate with factor B. So you're really shutting down this response. You also have CMCP, which is the membrane cofactor protein, and also uh, for factor I, the serine protease really cleaving your C4, really cleaving your C3, CB, C4B, C3B. Factor H is present because it binds to sialic acid residues. These are very abundant residues on your cells, on your host membranes. And they also cause dissociation. Factor H can cause dissociation of these factors once they bound to the cell as well. And in a similar way, your MCP is important for decay accelerating activity. So it causes the decay of these factors from binding, from being able to form these complexes on the cell membrane. In a similar manner, you also have decay accelerating, important for the decay of the complement system of the cell membrane. And then finally, you have properdin. Again, it's able to stop the association of the MAC complex. So if you're able to run through the entire complement system, able to activate your factors, your C7, C6, C7, and bring in C8, what this will stop is stop C9 associating with forming the cell. So you're able to shut the complement system.
Now, I think you explained that very well. We look at the deficiencies within the
Yeah. So if you have deficiencies in really in any of its complement proteins, it will bring up a vast array of clinical presentation. And so, but they will all do it if you have C1 to C4, will have something called an immune complex disease. Here, you really cannot form B3B, any of your C3B. So you cannot form anything really with your complement system. So you will kind of just get you'll kind of this complement system in general to these immune complex diseases. They'll kind of bundle up and they won't really go anywhere. You won't be able to take this process anywhere. C3 in particular, for pyogenic bacteria, these are uncapsulated, so you have access to that cell wall. Issues within the complement, within the alternative complement pathway. So if you're not able to spontaneously utilize C3, not able to get these spontaneous reactions occurring as your first line, you cannot really target these pyogenic bacteria. C5 to to envelop pathogens. So if you don't have the ability to attack these gram-negative bacteria, these enveloped ones, you're really susceptible to diseases from them. So if they are to infect, you cannot use infections is what you are likely to be suffering from. Real complement and a little bit of the clinical picture that they come into there, a little bit of that scope. But I think it's also worth to mention some evasion strategies to kind of down-regulate the complement system. A variety of these for evasion. But I think it's important to think of it through concepts. So the first thing complements to interfere with some of these complexes forming in this way. You won't be able to bind your C1q and initiate the classical pathway. Also, inactivating complement proteins. You have your own serine proteases if um, the pathogen proteases to start cleaving any C3 to its surface. Inactivating it, the cascade stops. You can't reach the C3 threshold to start the system. In a similar way, instead of just inactivating it, you just destroy it. So destroying complement proteins, really shutting down that part of the system. And then finally, if you're able to utilize their surface to really play the complement system, so it doesn't attack your own tissue. So you're not really getting your autoimmune diseases from complement deficiencies. If you have deficiencies in your complement regulator, I think I want to mention, you start seeing these autoimmune like conditions because your complement system is attacking the cells. So if your pathogen can utilize this mechanism by forming your regulatory factors, the complement system in that way. I'll mention Staphylococcus aureus. It has a specific protein, and formation on the cell membrane, and it kind of inactivates three and stops that process going downstream and the complement on the pathogens. So I think that is really of worth to know.
By what mechanism would they be able to destroy the complement proteins?
As I mentioned, if you have any protease activity, so is a hydrolytic enzyme able to cleave those tight bonds. If you don't cleave it, then it's not the C3b not able to associate, let's say, with C2a, your BB. So if you have your classic C5 convertase or your alternate C5 convertase, if you have your C3 convertase present on, but you start inactivating aspects of it, it's not able to form a C5 convertase, so you can't bring that in onto your membrane. Also, if you're able to bind it in a way to cause a conformational change, you will have this thioester group, very reactive group. If you're able to inactivate the thioester group through a conformational change, it won't really be on the surface. So in that way, onto the surface of
Parts of the protein. Right.
yeah, that's often what it is utilized for stories
And how do they inactivate it specifically?
through the cleaving process,
Mm.
through cleaving the complement.
And one more thing, could you provide us with some autoimmune disorders that can arise from problems within the complement system?
So, so kind of similar note, not really associated with this because these are autoimmune-like disorders. You're not using your adaptive immune system, right? So term that will be present throughout your life, really. So, yeah, present long. An autoimmune-like disorder because you're breaking down your own tissues, right? Through complement activation on host cells. You don't have this adaptive. Maybe we can link it to systemic lupus, SLE.
What are these three mechanisms? Let's explore them in detail.
So the complement system acts in sequential pathways. The first is the old reconfirmation, reacting with water in this tick over step, and they combined factor binding undergoes another conformational change, able to cleave it to form C. This will be cleaving it, and then the C3b is able to bind to the pathogen surface. Once on the surface, C3b again in the same process able to bind BB. Now your surface bound convertase, where it will, well, it's the alternative C3 convertase, it could have more C3 to form C3b, and then this accumulate on the pathogen surface and is involved in a positive feedback of binding more C3b to the surface with the reactive thioester group that gets exposed upon cleavage, and then again activating and factor B and cleaving, able to form more convertators, amplifying up the tagging of phagocytosis.
Rest the group you said. Can it the pathogen's surface?
It's very reactive. It's for binding to the pathogen surface to of the C and all these smaller molecules. They have an and they act as toxins and are able to communicate with other immune cells, especially phages, and other sentinels, so dendritic cells, not particularly, but basophils, eosinophils, roles in to complement receptors, macrophages, to be
So, if you want to learn more about the phagocytes, be sure to check out our other video discussing the innate immune system. Which mechanism you just covered?
I've talked about the Lectin pathway, which is really the first mechanism. It comes about from spontaneous conformational changes in C3 to activate this thioester group. Of C that there's just free roaming that happens on its own. So it's the first act, but then we have two more mechanisms that act sequentially. Step is the lectin pathway, which is a pathogen recognition receptor. Surfaces of microbes, different bacteria, fungi, even some viruses. And this soluble mannose-binding lectin is associated with MASPs. These are serine proteases, so BL binds to fructose or mannose residues, cleave C4 to A. Smaller fragment, usually the bigger fragment is used, but the C4b C2A is then able to C4b C2A is your classical C3 convert classical, and again you see this the classical part, the classical pathway, the next thing that we would explore.
1 second. What is this cleaving process?
You have your serine protease, for example, the MASP1 and 2, or if you have E4B, C2A, or your pathway convertase, these cleave your C3 or other proteins at specific residues. Your serine proteases, for example, triad of histone 57, the amino acids at certain points within the amino acid chain. And what this triad does is to cleave peptide bonds. Either, for an example, if you're able to cleave C3, you're able to 3B to then opsonize to pathogens.
Some examples of enzymes that cleave: the trypsin, chymotrypsin, kallikrein 4. Let's explore the classical form.
The classical pathway is this bridge between the adaptive and the immune system. If you're using antibodies against a certain pathogen, then is what really activates the classic. This is how you're able to use your innate immune system to really clear out pathogenic infections. So antibodies are able to pathogen surface. Also, you have C this pathway as well. Your antibodies bind to the pathogen surface, and then you have C1q, your complement protein that kickstarts this pathway, binding to the Fc region of the antibody. It has C1R and serine proteases, which then cleave C4 into C4b. Again, in the same way as the lectin pathway, C2A is then C2 is cleaved to C2A, which binds to the C4b, and then you're able to start cleaving C3 into C, and then this is your class. Then, if you have your C4A, C2B, it associate with a C3b, you're able to have a classical C5 MAC complex formation, the membrane attack complex formation, and be able to start causing the cell lysis part of the convertase, which is C3BB, C3, or C3B2, because you have two C.
Important question. The functions that we mentioned at the start of the video, where are they present in these three pathways and how do they overlap?
Yeah, so I think it's worth noting that the whole complement threshold, if you're able to have that are breaking down the complement system, you have to start promoting the complement system. It's really the stability that you're able to kind of build up C3. These effective functions. The different functions come from first, if you cleave your C3, you form B, but you also have a small component C3A. Your C3A is then acts as an anaphylotoxin, which is able to phagocytic cells to come to opsonize, phagocytose the pathogen. Your complement system, by forming your C3b on the cell surface, again, C3b receptors, for example, complement 1 is able to bind C3b but directly and is able to obsolete other small components from other complement proteins, such required to recruit macrophages, and it has to bind to a specific complement receptor on the macrophage for it to really opsonize the pathogen.
You said immune cell recruitment in all three pathways? Is it in a specific one?
Well, yeah, the immune cell recruitment is a system for, especially for the opsonization of these three pathways. And yeah, so you're forming these smaller fragments every time, especially because that's the main one you clean, but even other proteins. So definitely, yeah, in all these pathways you have activation and recruitment of phagocytic cells.
Okay. Now, should we look at the deficiencies associated?
Immune complex disease itself. The thing is, it has an adaptive immune form of these immune, especially uh, your tumors, the brain, and you start seeing those presentations. But yeah, it's very important to note that immune antibodies or autoreactive antibodies.
Okay. I think we've covered just about everything in the video. I hope you guys enjoyed and learned a lot about the complement system. Be sure to check out other videos discussing the innate and adaptive immune system and subscribe, to stay tuned for more podcasts like this. Thank you.